Good morning, everyone. Good morning. Yes, good morning. Good morning, everyone. Whether I'm audible? Whether I am audible? Yes, sir. Yes. 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 Today we are going to see the revision class for photosynthesis. Okay. Today we are going to cover photosynthesis, mineral nutrition, photoperiodism and vernalization. Okay. Before going to the lecture itself, I am saying sure you will get a minimum 4 to 5 questions from photosynthesis. Okay. So you have to listen the class properly. Just we are going to see the important things in photosynthesis. What are the important things and what are the questions they have already asked from photosynthesis. Okay. Then from mineral nutrition, sure we will expect 1 or 2 questions, and from photoperiodism and from vernalization, each topic one questions they will ask. Okay. So on an average, minimum 10 questions will be come from this lecture. Okay. So 10 questions will be we can expect. Okay. So listen properly. If you have any doubt, kindly let me know. Okay. First, we are going to see the basic definition of photosynthesis. Okay. First, what is meant by photosynthesis? It's a light-dependent anabolic process. Okay. Photosynthesis is a light-dependent anabolic process. Okay. Wherein the photoautotropic organisms. Photoautotropic organism means what? That have the capability of producing their own food by utilizing the energy from sunlight. Okay. That are autotrophic organism. Primarily example what will be there? Plants, algae and cyanobacteria that will convert inorganic carbon dioxide and water into energy rich organic compounds. Particularly if you see glucose molecule will be produced. Okay. The most important thing what you have to remember means during photosynthesis, light energy will be converted into chemical energy. Okay. This is the key point you have to remember. Okay. During photosynthesis, light energy will be converted into chemical energy, okay, and stored in the form of glucose molecules. Okay, this is most important thing you have to remember. Okay, then the overall equation of photosynthesis. This is the overall equation of photosynthesis. If you see, 6 molecule of carbon dioxide and 12 molecule of water molecule will be utilized to form a molecule of glucose. Okay, glucose will be formed, then oxygen molecules will be released. This is most important thing. Okay. Oxygen molecule will be released. Okay. Apart from that, 6 molecule of water will be recycled. Okay. So photosynthesis, okay, nothing. It's an oxidation. It's an oxidation of CO2 and reduction of carbon dioxide. Okay. These things also you have to remember. Okay. Then 6 molecule of water are oxidized during photolysis of water. This also we are going to see. Okay. There are 2 different phases in photosynthesis. That also we know. Light reaction will be there, dark reaction will be there. That also we are going to discuss very briefly in lecture. Okay. Then next most important thing what they will ask means scientist contribution. From scientist contribution, sure you will get 1 question. Out of 5 question from photosynthesis, you will get 1 question from the scientist contribution. Okay. So you have to listen properly. Okay. Van Helmont, he is the person who has given that water is one of the essential source for plant growth. Okay. He is the person who has conferred that water is an essential source for plant growth. Okay. Stephen Hales, father of plant physiology. Stephen Hales, okay. What he has concluded means light is essential in plant nutrition. Okay. Light plays an essential role in plant nutrition. Then Joseph Priestley, this is JRF 2022 question. Okay. Joseph Priestley. Okay, Joseph Priestley, what he has concluded means plant will release a gas molecules during the photosynthesis process. At later stage, they have discovered that gas is oxygen molecule. I hope this is clear. This is JRF 2022 question. Okay, Joseph Priestley, he is the person who has concluded that oxygen molecule will be evolved during photosynthesis process. Okay. Jan Ingenhouse, he has proposed photosynthesis is an light dependent process. Okay. Photosynthesis is an light dependent process. Okay. Then Nicholas Theodore de Sasser, okay, he has proved the stoichiometric clarity of the reaction. In previous slide we have seen 6 molecule of carbon dioxide will bind with 12 molecule of water to form the glucose along with oxygen molecule and water. This stoichiometric reaction was given by him. Okay. Then Julius van Sachs, he has proven the photosynthesis is happening in chloroplast. The organelle where the photosynthesis is happening in plant is chloroplast. This is also another important objective point of view. Okay, you have to remember properly. Okay. Now we are going to see the organelle, about the organelle. Okay, we have known photosynthesis will happen in chloroplast. Now we are going to see what is the arrangement of chloroplast, what are the things will be there in chloroplast. Okay, this is another important point. Okay, chloroplast is the photosynthetic organelle. This we have seen. Okay, the most important thing, it's a double membrane bound organelle. Okay, double membrane bound organelle. I am drawing here also. It's a double membrane bound organelle found in green plant cells. This will be responsible for capturing the light energy and converting it into chemical energy. We are going to see how the light energy will be captured, how it will be converted into chemical energy, everything we are going to see. Okay. If you see the shape of this chloroplast, it will be discoid or ellipsoidal in shape. Okay. Then if you see the size, it will be 4 to 6 micrometer in long, and if you see the width, it will be 1 to 2 micrometer. Okay. This you have to remember. Okay. Then the most important thing, the width of this two membrane, the width of this membrane will be 50 Armstrong will be there. Okay. The width of the two membranes will be 50 Armstrong. This is just for your understanding. Now we are coming to the most important thing. Okay. Like mitochondria, this space we will call as what? This is stroma, correct? This is stroma. Okay. Chloroplast stroma. This stroma is the primary site for dark reaction. This is most important objective point. Okay. So dark reaction will happen in stroma of the chloroplast. Okay. Then where the light reaction will happen? If you see, there will be a thylakoids, a stack of grana will be there, that we will call as thylakoids. Okay. This thylakoid is the major site for light reaction. I hope this is clear. So light reaction will always happen in where? In grana of thylakoids. Okay. Dark reaction, that is photosynthetic CO reduction cycle, that will be happen in stroma of the chloroplast. I hope this is clear for everyone. And most important thing, the chloroplast, like mitochondria, it's a semi-autonomous organelle. Okay. This point also you have to remember. It's a semi-autonomous organelle. Why we are saying it's a semi-autonomous organelle means it have its own genetic material, it can transcribe, then it will translate and produce its own proteins also. Okay. That's why we are saying this is a semi-autonomous organelle. Okay. Then you just see we have given the things, the grana, about the grana and the grana. Grana is the major site for light reaction. This we have already discussed. Okay. Then another most important thing, if you see in chloroplast, grana will be arranged. That grana will be arranged like this. Okay. How the granas, how the one and each other granas will be connected? This will be connected through stoma lamella, or we can call as fret membrane. So fret membranes primarily, what's the role means? The unstacked membranes will be connecting the grana. This is known as stoma lamella. Okay, stoma lamella. You just see this figure now. We have already discussed everything. Double membrane layer will be there. Okay. Then 50 Armstrong width will be there. And the most important thing, stoma. Stoma is the site for dark reaction. Then grana thylakoids will be there. This is the site for light reaction. Then fret membranes are there that will connect the one thylakoid membrane with another thylakoid. These are the basic things you have to remember about the structure of chloroplast. Okay. From here, we can expect 1 or 2 questions. Okay. Now, we are going to see the major sites. This we have already discussed. Stoma, site for dark reaction will be there. This we have already discussed. Okay. Now we are going to see how the light energy will be converted into chemical energy. Which molecule is responsible for absorbing the light energy? These things we are going to discuss now. Okay, these things we are going to discuss now. This is just a brief things. They have given the different membranes and where the reactions will happen. Thylakoid membranes, light absorption will be happen. Grana, this is the site for PS2 activity. Stroma lamella, PS1 activity will be more. Then stroma CO2 fixation will be happen. This we have already seen. Okay. Now we are going to see which pigments will absorb the light energy. Okay. How the light energy is absorbing. Okay. If you see the pigments, there are three different classes of photosynthetic pigments are there in plants. Okay. You observe the things, three things are there. One is chlorophyll, second is carotenoids, and third is phycobilins. These are the three different classes of plant pigments, that is photosynthetic pigments. Okay. We are going to see each and every class examples also, and what are the typical or critical points they will ask in exam point of view. Okay. So chlorophylls, this can be broadly classified into two types. One is primary pigment. Primary pigments means this will be directly involved in photosynthesis, that is chlorophyll A molecule. This is another important objective. Okay, chlorophyll A. Okay. Then accessory pigments are there. Accessory pigments is chlorophyll B, C, D and F. Okay, these all are accessory pigment. Apart from chlorophyll A, all other pigments are accessory pigments. So chlorophyll A is the primary pigment. Okay. Then we are coming to the second class, carotenoids. Okay. This carotenoids will be classified into two types. Okay. One is carotene, another is xanthophylls or carotenols. The another name is important. Xanthophylls or carotenol. Okay. And the third class we can call as phycobilins. Okay. Phycobilins. Here we have given the structure. What is the key difference between carotenoids, chlorophyll and phycobilins? This is very important. Here the question what they will ask means, among the plant pigments, among the photosynthetic pigments, phycobilin is the water soluble pigment. This is very important point. Okay, phycobilin is the water soluble pigment, but chlorophyll and carotenoids are insoluble in water. Okay, insoluble in water but soluble in organic solvents like ether. Clear? So, among the pigments, phycobilins is the water soluble pigment. Okay. Now, we are going to see the main characteristics of chlorophyll. Chlorophyll, that is the main pigment responsible for capturing light. This we have already seen. This is insoluble in water, but it can be extracted using organic solvents. This also we have seen. This will be primarily found in thylakoid membranes of the chloroplast. We already discussed thylakoid membrane is the primary site for photochemical or primary reactions. Okay. So, light reaction will happen in thylakoid. So, the chlorophyll will be present ultimately in thylakoid membranes. Okay. This, then the types, chlorophyll A, this we have already seen. It's the primary pigment, and chlorophyll B, C, D and F. These are accessory pigments. Okay. Coming to the carotenoids, carotenoids we have already seen two different classes are there, carotene and carotenols. Okay. Carotenols we can also say as xanthophylls. Okay. The main important thing is it is insoluble in water. Okay. It is insoluble in water. Okay. Now they have given the structural classification. Okay. The chlorophyll structure was first determined by Willstatter. This is another important objective. Okay. Willstatter and Hans Fischer elucidated the structure of chlorophyll molecule. That was confirmed by Woodward. I hope this is clear. Okay. So, the structure of chlorophyll molecule was elucidated by Willstatter and Hans Fischer. Then it was confirmed by Woodward. For this work, they have got Nobel Prize. Okay. For this work, they have got the Nobel Prize. If you see the basic structure of chlorophyll molecule, we know 4 tetrapyral ring structure will be there. Center magnesium atom will be there. This is the most important thing. In center, magnesium atom will be there. Then 4 tetrapyral ring structure will be there. At 4th ring, there will be an phytol chain. That phytol chain will be made up of 20 carbon atoms. This is also another important point. Okay. 20 carbon phytol chain will be added at the tetrapyral ring structure. Okay. And center atom will be magnesium. This is the key point you have to remember. It is a magnesium bound compound. Okay. Magnesium will be there in the middle. Then we see further classification of chlorophyll. Chlorophyll A, B, C, D, like that different classification is there. Okay. This they have asked in JRF 2021. Okay, 2021. What is the formula for chlorophyll A and chlorophyll B? Okay, formula for chlorophyll A and chlorophyll B. Okay, molecular formula for chlorophyll A is what? C55H72 O5N4 Mg. Why it is coming H72? Because it will have a methyl group at carbon 3 of third chiral ring. Okay, methyl group is there because of that extra 2 hydrogen atom is there in chlorophyll A. But if you see in chlorophyll B, the formula is C55H70 O6N4 Mg. Okay, you have to observe here. Here O6 is there, 2 molecule of hydrogen will be reduced. Because why? Aldehyde group will be present. In chlorophyll B, aldehyde group will be present. Okay, then we have already seen chlorophyll A is the primary pigment. Okay, that is the primary pigment involved in photosynthesis. Apart from chlorophyll B, everything will be an accessory pigment. Now comes the question, what is meant by accessory pigment? See, only chlorophyll A molecule will be involved in photosynthesis. Remaining whatever molecules are there, pigment molecule is there, that will absorb the light energy and it will transfer the light energy to chlorophyll A. Okay. This phenomena we can call as what? This phenomena is known as Foster or resonance energy transfer. Okay. So whatever the pigment is there, that will absorb the light energy and it will transfer the light energy to chlorophyll A molecule. Okay. I hope this is clear. Okay. Now we are going to see about other chlorophyll molecules. Okay. If they want to set the question paper difficult, they can ask about chlorophyll C and D also. Okay. The most important thing about chlorophyll C is this will lack the phytol chain. I already told phytol is a 20 carbon compound. So if you see this molecular formula for chlorophyll C, this will have only C35. Okay. In normal chlorophyll will have C55 will be there. Because of this lacking phytol chain, it will have carbon 35 only. Okay. So C35. Wherever C35 is coming, that will come as chlorophyll C molecule. Okay. In chlorophyll D, in spite of OCHO group, CHCH2 group will be there at second chiral ring. Okay, second chiral ring. This is the important things about chlorophyll C and D. And most important and repeated question, another question. PO15. Okay, PO15. This structure will be similar to chlorophyll A molecule. Okay, except the thing is it will lack the magnesium atom at the middle. At the center, magnesium atom will be there. This will not present in chlorophyll A molecule. Okay, this is another important and repeated question. So, in PO15, central magnesium atom will not be there and that magnesium atom will be replaced by 2 hydrogen atoms. That magnesium atom will be replaced by 2 hydrogen atom. Then what is the function of this PO15? It will act as an electron transporter during photosynthesis. This we will see in Z scheme. Okay, while discussing the Z scheme, I will tell what is the exact function of PO15. Okay. Next, we are going to see about carotenoids. Okay, carotenoids, this we have already discussed. Okay, 2 classes will be there. Carotenes and carotenols, like the 2 structure will be there. 2 classes will be there. The most important thing, they can ask the general formula or molecular formula for carotenes. Carotenes means C40H56. I hope this is clear for everyone. Carotene means C40H56. And most important thing, it is an open chain of conjugated double bonds. If you see in chlorophyll molecule, it will be ring-like structure will be there. But this is an open chain of conjugated double bond molecule. Okay, this is another important point you have to remember. Okay, and the most important function of carotenoid is what? It will prevent photodynamic or phytodynamic damage. Okay, this is the most important function of this carotenoids. Okay, what it will do means when excess of light will be absorbed by chlorophyll molecule, this excess of light may leads to ROS formation that may destruct the chloroplast membrane. In that case, these carotenoids will help in preventing the phytodynamic damage or photodynamic damage. I hope this is clear for everyone. Here they have given the different examples of carotenes. Carotenes, alpha carotene, lycopene, these are the different examples. Okay. We have seen about carotenes. Okay, now we are going to see about carotenols. Okay, carotenols, CAROTE, carotenols. Okay, so carotenols we can also call as xanthophylls. Okay, I hope this is clear for everyone. Carotenols we can also say as xanthophylls. This is another important thing you have to remember. Okay, oxygenated carotenes are known as carotenols. Okay, oxygenated carotenes are known as carotenols. We know carotene formula C40H56. If oxygen molecule is added here, that we can say as xanthophylls or carotenoids. I hope this is clear for everyone. Okay. Now these are the some of the examples for xanthophylls. Viloxanthine, lutein and zeaxanthin are some of the examples. Okay. Then the role of carotenoids, this we have already discussed. Role of carotenoids, the primary role is what? It will prevent the photosynthetic machinery from damage caused by reactive oxygen species. Okay. So ROS mediated damage can be prevented. That is photodynamic damage can be prevented by carotenoids. I hope this is clear for everyone. Up to this, there is no doubt, I hope. Any doubt is there up to this? Any doubt, students? Any doubts? Safura, any doubt? Svetaraj, Surabhi, any doubts? If no other doubt means I will proceed. Okay. Then the third molecule is phycobilins. This we have already seen. Phycobilins is an water soluble molecule. Yes, water soluble molecule, and the most important thing, it will be red and blue white pigment. This is red and blue white pigment. Critically, this will be found in cyanobacterium and red algae. These are the points you have to remember. Okay, don't go and read much. These are the key points you have to remember. Okay. Now we have seen what is the molecule that will absorb the light energy, that we have already seen. Okay. So chlorophyll, carotenoid and phycobilins are the molecule that will help in capturing the light energy. Okay. Now we have to see what is the source of that light energy. If you see, sun is the primary source of the light energy. This is very simple question. Okay. Now question what they will ask in this is, if the chlorophyll molecules are whatever the plant pigments is absorbing the light, how many percent of the absorbed light will be utilized for photosynthesis? This is JRF 2021 question. Okay. JRF 2021 question. Okay. So out of the total energy absorbed by the molecules, only 1% will be actually utilized in photosynthesis. I hope this is clear for everyone. Only 1% will be utilized for photosynthesis process. Okay, only 1%. Okay. Then another most important thing, what is photosynthetically active radiation? PAR. Okay. The range they may ask. Okay. So these chlorophyll molecules, these molecules will not absorb light at a random wavelength. Okay. It will absorb light only at the specific wavelength, that is known as photosynthetically active radiation. PAR. Okay. That range will be 400 to 700 nanometer. This is another important point you have to remember. Okay. This is the different example. For example, if you see chlorophyll A, B, C, D is there, then bacterial chlorophyll is there. They have given the absorption maximum at in vitro and in vivo. You have to remember only for chlorophyll A and B. This is very important point. You have to remember at in vitro condition and at in vivo condition, what is the absorption maximum for chlorophyll A and chlorophyll B? This you just go through once. Okay, this they may ask. Okay. Next thing, we are going to see how the energy absorbed by the accessory pigment will be transferred to the chlorophyll A molecule. This we have already seen. Chlorophyll A is the primary pigment absorbed directly in photosynthesis. Whatever the energy that is absorbed by the accessory pigments will be transferred by the process known as resonance energy transfer or Foster mechanism. By this process, absorbed light energy will be transferred. I hope this is clear for everyone. I hope this is clear. Okay. Now we are going to see what happens when pigment absorb light. If a chlorophyll molecule or whatever the pigment molecule is there, if we observe the light, what will happen there? Okay, what it will happen to the chlorophyll molecule? This we have already seen. Okay. If a molecule is there, if we are giving some light energy for some energy, it will go to excited state. Okay, it will go to from ground state to excited state. It will go. Okay, then it may lose the energy, whatever light energy it was observed. This may released and it will come to the ground state. This is the basic. Okay, this is the basic. Now we are going to see how the plant pigment, how the photosynthetic pigment, if it absorb light, what is the mechanism will be happen there. Okay, if a chlorophyll molecule is there here, just to imagine, if a photon is passing here, it will go to excited state. Okay, excited state it will go. Okay, that we can call the singlet state. Yes, is singlet state. This is very important point you have to remember. Okay, it will go to excited singlet state. It will go. Okay, then by the process of vibration, it will release some of the energy. Okay, the singlet state time is 10 power minus 12 second will be there. Okay, that is singlet state. Okay, then by the process of fluorescence, from the process of fluorescence, it may release the extra energy. So, by the process of fluorescence, it will release the excess of energy, then it may come to ground state. This is process 1. Okay, clear? This is process 1. Then second stage is there. If some of the energy will be released as internal crossing over, it will come to another stage. This is known as metastable state. Metastable state. Metastable or triplet state. This is another important thing you have to remember. Triplet state. This is 10 power minus 3 second. This is very important. Okay. This is very important. Okay. So from here, it can lose some of the energy. Then it can come to the triplet state, or we can also say as metastable state. That time is 10 power minus 3 second. Okay. This is the energy state. This is the energy state where the energy will be utilized for the light reaction process. So 10 power minus 3 second is the most important thing. Okay. From here, how it will come to ground state means by the process of phosphorescence. I hope this is clear. Okay. So two phenomena are there. When a chlorophyll molecule absorbs the light energy, it will go to excited state. Okay, that is known as singlet state. From singlet state, if it has to come to ground state, it can follow the process of fluorescence. Clear? Then from metastable state or triplet state, it has to come to ground state, means it will come in the process of phosphorescence. Okay, these two are very important phenomena. This they can ask in assertion, reason or statement question. Okay, this they can ask. Okay, so you have to remember properly. These things we have given here, how it will happen like that. Whatever we have discussed, that thing only we have given here. And the most important thing what you have to remember about this fluorescence and phosphorescence is, you have to remember the wavelength of the emitted light will always be longer. Okay, that will be always be longer. So the wavelength of the emitted light will be always longer. Okay, in both fluorescence as well as in phosphorescence. Okay, this is another important point you have to remember. Okay, clear? Now we are going to see the simple concept. What is meant by quantum yield, quantum requirement. This is JRF 2023 question. Okay. JRF 2023, they have asked definition. They asked what is meant by quantum requirement, what is meant by quantum yield. Okay, we have already discussed during the process of photosynthesis, oxygen molecule will be released. Okay, so quantum requirement means number of photons required to release a molecule of oxygen. This is clear. So the number of photons required to release a molecule of oxygen is known as quantum requirement. If you see, around 8 to 10 quantums are required. This is also another important point. 8 to 10 quantums or 8 to 10 photons are required to release a molecule of oxygen. Okay, clear? So quantum yield means what? Per photon. Okay, per photon, how much molecule of oxygen is releasing? Clear? So number of oxygen molecule released per photon. So what will be the answer? 1 by 8, around 0.8 will be there. Okay, 0.8 will be there. Okay, clear? So quantum requirement and quantum yield definition is very important. Quantum requirement means number of photons required to release a molecule of oxygen. Quantum yield means number of photons that will release a molecule of oxygen. Okay, this is very important point you have to remember. Okay, another important thing, we have already discussed these things. There are two main phases are there. One is primary photochemical reaction that we can call as light reaction or Hill reaction we can also say. Then dark reaction we can also say as Blackman reaction or path of carbon photosynthesis. Okay, path of carbon synthesis. Okay, this we have already discussed. Simple outfit motivation chan. Good night. Don't do like these commands. Huh? If you don't want to listen the class, just you quit from here. Okay. So two main phases are there. One is primary photochemical reaction. Second is dark reaction. Okay. This we have already discussed, light reaction and dark reaction. Okay. Where it is happening? Light reaction will happen in thylakoid membrane. Okay. Light reaction will happen in thylakoid membrane. Okay. Dark reaction will happen in stroma of chloroplast. Okay. Dark reaction will happen in stroma of chloroplast. It will happen. Okay. So in light reaction, what it will happen means light energy. By using the light energy, ATP and NADPH will be produced. Okay. This is the primary function of light reaction. ATP and NADPH will be produced. In dark reaction, whatever the ATP and NADPH is produced, that will be utilized to convert the carbon dioxide to sugar molecules. That means reduction of CO2 will be happen there. Clear? So these are the major function of light reaction and dark reaction. Now we are going to see light reaction. Okay, how the light reaction will happen, then how the dark reaction will happen and what are its classification. Simple outfit motivation chan. Okay, sorry sir. Okay. See, we are teaching these things for your benefit, not for our benefit. Okay. Don't do these comedy things in class. If you don't want to listen, please get out from the class. Okay. Next thing. Okay. Now, two pigment system are there. We have already discussed in light reaction what it will happen. Yes, in light reaction what it will happen. We have already discussed. Okay. Now, how the light reaction is happening, we are going to see. Okay. First, the light reaction will happen in where? Thylakoid membrane, that we have already discussed. Okay. So, thylakoid membranes will have photosystem. We know two different photosystem will be there. PS1 and PS2. Here the question what they will ask means PS1 is reaction center that is having P700. P700. Okay. Then PS2 means what? Reaction center will be P680. Okay. P680 and P7. These are the key question they will ask. So photosystem means what? It's a protein pigment complex. We know the chlorophyll molecule, everything will be there. These are the pigments. Along with that protein will be there. These two complex will form photosystem. Okay. I hope this is clear for everyone. Okay. So the primary photochemical reaction will be always faster than the dark reaction. This also you have to remember. So primary photochemical reaction will always be faster than the dark reaction. Okay. Now we are going to see the sequence of reaction. What are the reaction will happen in light reaction? Okay. These are the steps. First, absorption of light energy will be done by the chlorophyll molecules. Okay. First, light energy will be absorbed. After that, whatever the light energy is absorbed by accessory pigment will be transferred to chlorophyll A molecule, because we know chlorophyll A is directly involved in photosynthesis process. Okay, that is light reaction. Okay. Then activation of chlorophyll molecule will happen by photon of lights. Then most important thing, photolysis of water will happen and oxygen evolution will be done. After that, electron transport and production of assimilatory power will happen. These are the basic steps we are going to see briefly. What is what? Okay. So this is clear. First, light will be there. If light is falling on the molecules, that is pigment molecule, okay, the light will fall on the pigment molecule. It will excite to the excited state. Okay. This energy will be, all the energy will be transferred to chlorophyll A molecule. Because of transferring the energy to chlorophyll A molecule, this will be primarily involved in photochemical reaction. Because of this, what it will happen means assimilative powers will be produced by the process of electron transport chain. Okay. Photosynthetic electron transport chain will happen. Because of that, ATP and NADPH will be produced. This we are going to see briefly. Okay, this we will see here briefly. Okay, these are the different things whatever we have discussed that they have given here. Okay, chlorophyll A is the primary pigment, remaining are accessory pigment they have given here. What is photosystem 1, what is photosystem 2, this also we have seen already. Okay, the most important thing I will say you. Okay, then oxidation of water will be happen. Okay, why the oxidation of water will happen, everything I will show in diagrammatic way. This just for your observation. Just you see one, photolysis of water will happen, oxidation of water will happen, that things I have given it. Okay, now the most important thing, these all are like simple things only you have to see. Okay, now the most important thing, this is the most important thing you have to remember, because JRF 2019, 22 and 23, 3 years continue, this 3 years they have asked this question. Okay, from this part they have asked 3 years question. Now we are going to see the mechanism and how it is happening. I already told, because of this light energy absorbed by the molecule, ATP and NADPH will be synthesized. Okay, there are two different ways of synthesizing the ATP and NADPH. One is cyclic phosphorylation and second is non-cyclic photophosphorylation. Okay, photophosphorylation means what? It's synthesis of ATP and NADPH2. Okay, during the process of photosynthesis. Okay, so first we are going to see non-cyclic photophosphorylation. Okay, in non-cyclic photophosphorylation, the most important thing you have to remember is both pigment system will be involved. Both the pigment system will be involved. This is the first point you have to remember. Two pigment system will be involved. Okay. Then second most important thing you have to remember is, see, first light is falling on the PS2. It is going to the excited state. The electron will be captured by, this is the most important thing. PO15 is the first electron acceptor. PO15 is the first electron acceptor from PS2. This is another important point you have to remember. Okay. Then the electron will be flow through electron transport chain, that is plastoquinone will be there, cytochrome B6F will be there, apart from that plastocyanine will be there, then it will reach to PS1. Okay, then it will reach to PS1. Here what they will ask means, plastocyanine is a copper containing membrane bound protein. Okay, so plastocyanine is a copper containing membrane bound protein. Okay, so if you observe here, the electron is released from this PS1, PS2, but because of this, electron hole will be happened here. To fill that electron hole, photolysis of water will happen. Okay, because of photolysis of water, electron will be donated and the electron hole will be filled in PS2. I hope this is clear. So PS2 is the site for photolysis of water. Okay. In PS2, photolysis of water will be happened. This is clear. I hope so. Okay. Then from PS1, if again light will form, means electron will go to excited state. Here the primary electron acceptor is A0. Okay. So A0 is what? It is a phylloquinone. Phylloquinone. These are the key points they will ask. Phylloquinone. This is the primary electron acceptor from PS1. I hope this is clear. So PS1 primary electron is phylloquinone or A0 molecule, and PS2 the primary electron acceptor is PO15. Okay. From here the electron will be passed through to ferredoxin NADH reductase oxidoreductase complex. Because of this, NADPH will produce. If you see here, in non-cyclic photophosphorylation, that is also known as Z scheme, both the ATP and NADPH will produce. This is the another important point you have to remember. Both ATP and NADPH2 will be produced in non-cyclic or Z scheme of photophosphorylation. If you see cyclic photophosphorylation, only PS1 will be involved and the electron that is excited will be cycled back to the PS1. And the most important thing here, only ATP will be produced, that is 2 molecules of ATP is produced. If you see in non-cyclic or Z scheme, 1 ATP and 1 NADPH will be produced. NADPH2 will be produced. Clear? This is the number of ATP and NADPH molecules produced in cyclic and non-cyclic photophosphorylation. Okay, these are the key difference. Okay, cyclic electron transport chain, it will be primarily associated with PS1. both PS1 and PS2 will be involved in non-cyclic or Z scheme. Okay. The electron expelled from chlorophyll molecule is cycled back, that we have already seen. But here it will not cycle back because of this photolysis of water will take place. So photolysis of water is happening means oxygen will be released here. Here photolysis of water and oxygen evaluation will not be there. And another most important thing, photophosphorylation. That means ATP will be synthesized here two places, but here only one places ATP will be synthesized. And another most important thing, NADPH is not produced here, NADPH will be produced. I hope this is clear for everyone. With this we are completing light reaction of photosynthesis. Okay, we have started what are the different pigments, what is the definition of photosynthesis, scientist contribution, then light reaction we have completed. Any doubt so far? Any doubt is there? Kindly let me know any doubt is there. Whether you are understanding the class, any doubt is there? No doubt, okay. Okay, if we have, with this we are completing the light reaction, we are going to start now dark reactions. Okay. After completing dark reaction, we will take one break. Okay. Now we are going to see about dark reaction. Okay. Dark reaction is what? It is second phase of photosynthesis. Okay. So in dark reaction, whatever ATP and NADPH2 produced during the light reaction will be utilized to fix the carbon dioxide. Clear? So whatever ATP and NADPH was produced in light reaction will be utilized for CO2 reduction. Okay. CO2 reduction and CO2 fixation. Okay. And most important thing I forgot. Okay, in photosynthesis reaction, carbon dioxide will bind with water, 6 molecule, here 12 molecule will be there. That will lead to formation of glucose plus oxygen molecules plus water. Clear? So, here reduction of carbon dioxide will be happen. Okay, oxidation of water will happen. This is another important point. Okay. So that's why we are saying oxidation of water will happen and reduction of CO2 will be happen in photosynthesis. This is another important point. Okay. Now we are going to see about dark reaction. See, in dark reaction you have to remember three major pathways. One is C3 cycle, then C4 cycle and CAM pathway. Okay. These are the different pathways. These are the different pathways involved for CO2 reduction or CO2 fixation in the plants. Okay. Now we are going to see C3 pathway, C4 pathway and CAM pathway. What is what? What are the important point for exam point of view? Okay. C3 cycle we can also say as Calvin cycle. Okay. Or we can also say as C3 pathway or Calvin-Benson cycle or photosynthetic carbon reduction cycle or reductive pentose phosphate pathway cycle. These are the different names we are giving to C3 cycle. Why we are calling that C3 cycle as C3? Okay. If you see the first carbon, the first compound formed is 3 carbon compound. That's why we are saying that as a C3 cycle. Okay. What is the 3 carbon compound formed? Everything we are going to see. Okay. That is 3 PGA is the first 3 carbon compound formed. Okay. If you see this C3 cycle, there are 3 different stages are there. Okay. 3 different stages are there. First is carboxylation stage. Okay. Second is reduction stage. Third is regeneration stage. Just you remember, first is carboxylation. Second is reduction. Third is regeneration. Okay. What will be regenerated? Everything we are going to see now. Okay. So the first CO2 acceptor, the CO2 acceptor in C3 cycle is Rubisco molecule. Okay. Rubisco will be there. Sorry, RUBP will be there. Okay. RUBP is the first CO2 acceptor molecule. Okay. RUBP is a 5 carbon compound. Okay. It will bind with carbon dioxide. Okay. This is a 5 carbon compound. It will bind with carbon dioxide. The enzyme acting on this is Rubisco. Okay, Rubisco. Rubisco is the enzyme acting on it. From Rubisco, every year they are asking at least one question. You have to remember about the Rubisco. Rubisco is the most abundant protein, most abundant soluble protein. Okay. This you have to remember. It is the most abundant soluble protein. And another most important thing what you have to remember means this Rubisco will have L8 and S8 subunit. Total 16 subunit will be there in the proteins. Okay. This larger subunit will be synthesized from chloroplast. Okay. Smaller subunit will be synthesized from nucleus. This is another important objective point you have to remember. And another most important thing, how the Rubisco will be regulated. Okay, how the Rubisco will be regulated. After this C3 cycle, I will tell how the Rubisco will be regulated. So now it is clear. First carbon dioxide will be there, that will bind with RUBP, ribulose 1,5 bisphosphate. By the action of Rubisco, it will convert the processes. Okay, you see here, enzyme is Rubisco. First it will form a 6 carbon unstable compound. It will form. Okay, this 6 carbon unstable compound will be degraded to form 2 molecules of 3 carbon compound, that is 3 PGA, 3 phosphoglyceric acid. This is the first 3 carbon compound formed in C3 cycle. This is the key point. This is the question they are going to ask. Okay, so 3-phosphoglyceric acid is the first stable product of the cycle. I hope this is clear. The second phase is reduction. Whatever the ATP and NADPH2 that are produced in light reaction will be utilized in this reduction phase. You see here, in first step, ATP is utilizing. In second step, NADPH is utilized here. Okay. If you see here, in first step, 3-phosphoglyceric acid will be converted into 1,3-bisphosphoglyceric acid. This 1,3-bisphosphoglyceric acid will be converted into 3-phosphoglyceraldehyde. Okay, 3-phosphoglyceraldehyde will be formed. So, ATP and NADPH will be utilized in reduction phase. Third phase is regeneration. What has to be regenerated here? If we see in first phase, RUBP was utilized. Correct? RUBP was utilized for binding with CO2. This RUBP will be utilized. This RUBP utilized in the stage 3. In stage 3, the RUBP molecules will be utilized. Okay. This you don't want to see the reaction, how the RUBP is regenerated. Just you go through once. I have given here, okay, just you go through once. Okay, these are the reaction that are involved in RUBP regeneration. If you see this graph, it will be very clear for you. Okay. First CO2 molecule will come, RUBP will be there. Okay. Rubisco carboxylase activity will happen here. Because of the 6 carbon compound will form. This will be degraded to form 2 molecules of 3-phosphoglyceric acid. Then ATP and NADPH will be utilized. That will lead to formation of 3-phosphoglyceraldehyde. This 3-phosphoglyceraldehyde, this is two molecule will be there. This by the action of isomerase, it will be converted into dihydroxyacetone phosphate. This 2 will bind means 6 carbon compound will form, that is fructose 1,6 bisphosphate. Here if phosphatase will come means phosphate group will be removed, fructose 6 phosphate will form. Up to this clear for everyone, fructose 6 phosphate will form. This can be taken out for sucrose biosynthesis. This can be taken out for sucrose biosynthesis. If it has to be regenerated means fructose 6 phosphate will bind with another molecule of glycerol, another molecule of 3 PGA to form erythrose 4 phosphate and pseudoepiderose 7 phosphate. How means fructose is 6 carbon compound plus glyceraldehyde 3 phosphate is 3 carbon, total 9 carbon will be there. Okay, that has been formed like this. Okay, like this the regeneration reaction will happen. Okay, by combining these things, regeneration reaction will happen. The key point you have to remember here is regeneration phase. In regeneration phase, RUBP will be regenerated and extra ATP will be utilized. Okay. Now we are going to see the stoichiometry, how much number of ATP will be utilized. These are the key questions they will ask. How much number of ATP will be utilized for fixing a molecule of carbon dioxide. You have to understand the question whether they are asking for producing a molecule of exose sugar or fixing a molecule of CO2. These both are different. Okay, these both are different. Okay. If exose molecule has to produce means 6 molecule of carbon dioxide has to be fixed. I am right. If only 1 molecule of CO2 means we have to just divide by 6. Okay. So these are the key feature. You just see once location, stoma of the chloroplast it will happen. Light requirement, no, indirectly only it needed because ATP and NADPH will be there. Key enzyme is Rubisco. First product is 3-PGA. This we have already seen. End product glucose may be formed. Energy source is ATP and NADPH2. Cycle restart when the regeneration of RUBP will happen. This is easy for everyone. Okay. Now another most important thing you have to remember, the oxygen evolved during photolysis of water will comes from only water and not from carbon dioxide. Okay. The oxygen molecule released during photosynthesis process is coming from water molecule, not from carbon dioxide. This was proven by Van Neel. Okay. This is another important objective point you have to remember. Okay. The source of oxygen released in photosynthesis is from water molecule. This was proven by Van Neel. Okay. This is the stoichiometry, just you see. Okay. If you see here, a total of 12 NADPH molecule and 18 molecules of ATP will be utilized for fixing 6 molecule of CO2. Okay, for fixing 6 molecule of carbon dioxide, we need 12 molecule of NADPH and 18 molecule of ATP. Okay, for 6 molecule, they can ask per molecule of CO2 fixation, how much number of ATP will be utilized. Just you divide this by 6. Okay. Then they can ask how much number of ATP will be utilized for triose sugar formation. Okay. For triose sugar formation, that means you just have to divide this by, okay, you just have to, like, 3 carbon will be there for formation of triose molecule. I hope this is clear for everyone. From stoichiometry, they will ask one question. Okay. Then they can ask the example of each and every crop. What are the example of C3 plants? Like rice, wheat, like that crops are there. Na? They can ask the example of C3 plants also. These are the key points they will ask. Okay. Next, we are coming to C4 cycle. Okay. C4 cycle. This is another important point. Okay. I already told that Rubisco will have two activities, Rubisco carboxylase and Rubisco oxygenase activity. Okay. Before going to this lecture, I will like tell some of the important things about Rubisco. Okay, Rubisco. It will have both the carboxylase activity as well as oxygenase activity will be there. Okay. How it is regulating? Okay. How the Rubisco will be regulated? This is one of the important point you have to remember. Okay. If Rubisco, in Rubisco, lysine residue will be there. Particularly if you see lysine 201. If this lysine 201 molecule is carboxylated, okay, carboxylated, it will become active. This is another important point you have to remember. Lysine 201 residue carboxylated means it will become active. And second most important thing, Rubisco activase enzyme will be there. Rubisco activase enzyme. Okay. This will activate the Rubisco. This will activate the Rubisco. This is another important thing. By utilizing ATP, it will activate the Rubisco enzyme. Okay. These two are very, very important point. Then another important point, 1 year, what they asked means, what is the catalytic efficiency of Rubisco? Catalytic efficiency or turnover of Rubisco. Okay. If you see, only 3.3 molecule of CO2 will be fixed per second. Okay. Only 3.3 or approximately only 3 molecule of CO2 will be fixed by Rubisco per second. That's why Rubisco is most abundant. Okay. Photosynthesis has to be happen more. So more amount of that Rubisco has to be produced. Because of that, that is an abundant soluble protein in earth. I hope this is clear for everyone. Okay. That are the key points they will ask. Okay. Now the most important thing, we are coming to C4 cycle. Okay. C4 cycle. First, we are going to see who has discovered this C4 cycle. Okay. Hatchell and Slack. Hatchell and Slack has discovered this C4 cycle. And the most important thing you have to remember is C4 cycle, the wherever the plants happening C4 cycle, that is C4 plants, having Kranz anatomy. Kranz anatomy will be there. That is what means Kranz anatomy means the bundle seed cells will be there. This is bundle seed cells that will be enclosed by mesophyll cells. Why it is happening like this means to efficiently increase the concentration of CO2, particularly in the bundle seed cell. Okay. This arrangement of this chloroplast is known as Kranz anatomy. This they have asked in JRF 2023. Okay. What is Kranz anatomy? In which plants it will be there? This they have asked in JRF. Okay. You see here, C4 plants will have a specialized structure that is known as Kranz anatomy. Vascular bundles are surrounded by a ring of bundle seed cells. Okay. These are surrounded by mesophyll cells. Okay. Then the most important thing, this they have asked in assess and reason question. Okay. Chloroplast in bundle seed cells will always be large and lack granule. That means A granule. Okay, large and A granule. The chloroplast in mesophyll cells is small and it contains granule. This is another important point you have to remember. Okay, this is the arrangement you see here. The mesophyll cells is there outside. Bundle seed cells are arranged inside. Okay, the bundle seed cells will be large and A granule. This will be small and granule. These are the important thing you have to remember. Now we are going to see about C4 pathway. The similar thing only, whatever pathway we are studying, we have to know what is the first CO2 acceptor, what is the first product formed, what is the stoichiometry of ATP and NADPH utilized. These are the key point. Then fourth important point you have to remember, what are the examples and what are the regulatory enzymes present in that pathway. Okay. The first step of C2 like C4 pathway is CO2 will be converted into carbonic acid. Okay. CO2 will be converted into bicarbonate or carbonic acid. Okay. For example, if you see the diagram is there in next slide. Okay. Mesophyll cells will be there, bundle seed cells will be there. First CO2 will enter here. The CO2 will be converted into carbonic acid or bicarbonate. Okay. By the enzyme carbonic anhydrase. The cofactor for carbonic anhydrase is zinc. This is another important objective point you have to remember. So, zinc is the cofactor for carbonic anhydrase. So, first carbon dioxide will convert into bicarbonate. This will be bind with phosphoenol pyruvate. So, the first CO2 acceptor on C4 cycle is phosphoenol pyruvate. Okay, phosphoenol pyruvate. By the action of PEP carboxylase, it will convert to oxaloacetate. So, first product formed is oxaloacetate. It's an 4-carbon compound. It's a 4-carbon compound. That's why we are saying this as an C4 pathway. C4 pathway. Then what it will happen? This oxaloacetate will be converted into malate or aspartate. Malate or aspartate. That we will see. Okay. There are 3 variants of C4 pathways is there. Based on that variant, whether the oxaloacetate will be converted into malate or aspartate will be decided. Okay. That we will see. That also they have asked one time question. Okay. So next step in the C4 pathway is what? The malate will be moves out of the mesophyll cells and it will reach to bundle sheath of the chloroplast. There it will be decarboxylated. Okay, that will be decarboxylated there. That decarboxylated CO2 will be utilized. That decarboxylated CO2 will be utilized by Rubisco enzyme present in bundle sheath cell. Okay, Rubisco enzyme present in bundle sheath cell for CO2 fixation. You just have to see this figure. If you see this figure means everything will be clear. Okay, you just see this figure. This is the simplified figure. First CO2 will come. In mesophyll cell, CO2 will be converted into bicarbonate. This bicarbonate will be first accepted by phosphoenol pyruvate. This is the first CO2 acceptor. Because of this phosphoenol pyruvate, PEP carboxylase enzyme will come. This will lead to formation of oxaloacetate. This oxaloacetate will be converted into malate. This malate will go to bundle sheath cell. This we have seen already. This malate will go to bundle sheath cell. Here it will be decarboxylated. This CO2 will be utilized by Rubisco enzyme. This CO2 will be Rubisco enzyme. Then CO2 fixation will happen. This pyruvate will be converted back to phosphoenol pyruvate. This is the simplified figure. Here what question they will ask means, you have to remember here what question they will ask means, where the C3 cycle will happen in C4 plants. You understand the question? Where the C3 cycle will happen in C4 plants. In bundle sheath cell. In bundle sheath cell of C4 plants, C3 cycle will happen. I hope this is clear for everyone. Okay, in bundle sheath cell, C3 cycle of C4 plant will happen. This is very, very, very important question, not only for JRF, for ARS also they will ask this question repeatedly. Okay. Now we are going to see the variants. There are 3 variants I told. On what basis they have classified these variants? Okay, on what basis they have classified these variants? These variants are classified based on how the decarboxylation will happen, how the decarboxylation reaction will happen. We have seen here the malate is decarboxylated in chloroplast to form CO2. Okay, based on the mechanism how the decarboxylation is happening, they have classified these C4 plants into three types. What are the three types? I am just writing here, okay. NAD malic enzyme time, NADP malic enzyme type. Okay, NAD malic enzyme time, NADP malic enzyme type, and PCK type. PCK type. These are the three different types. Okay. NAD, NADP, PCK type. These are the three variants. In these three variants, what question they will ask means you have to remember the example. Example for NADP malic enzyme, NAD malic enzyme, and PCK type. What is the example? Where the carboxylation will happen, where the decarboxylation will happen. Okay. Remaining energy stoichiometry, everything will be same for these things. The key difference is where the decarboxylation will happen. Okay. I have simplified and I have given in this table. You just read this table, that is highly sufficient. Okay. NADP malic enzyme, it will, the primary site of CO2 release is chloroplast. Okay. The typical example for this is sugarcane and sorghum. Okay. NAD malic enzyme, the primary site for decarboxylation is mitochondria. Okay. Example is amaranthus and panicum repens. Okay. Amaranthus and panicum repens. And PCK type, the decarboxylation will happen primarily in cytosol, and the example is panicum maximum. Panicum maximum and panicum coloratum. These are the typical example for PCK type. You just have to remember what are the different types, where the site of CO2 release will happen, and example of plant. From here they have asked question. JRF 2024. 2024 is one of the difficult question. Okay. From there, difficult question paper, from there they have asked this question. Okay. Okay, clear? I hope this is clear for everyone. Now we are going to see the energy stoichiometry. Okay. In C3 cycle, for fixing 1 molecule of CO2, 3 molecule of ATP and 2 molecule of NADPH was utilized. In C4 cycle, per molecule of CO2 fixation requires 5 molecule of ATP and 2 molecule of NADH2. So this is what higher energy is required. Okay. Higher energy is required. Okay. High energy is required. I hope this is clear for everyone. Okay. Now we are going to see the last pathway, CAM pathway. Okay. CAM pathway, crustacean acid metabolism pathway. Here the key point, what they will ask means, that key points first I will tell, then we can go and discuss. So the key point is, CAM plants will be always found in dry, hot and humid conditions. Okay. So the water use efficiency of these CAM plants will be more comparing to C3 and C4 plants. This they have asked one time. So CAM plants will have higher water use efficiency. Why it is having higher water use efficiency? That we have to know. Okay. These CAM plants will open their stomata only during night time. Okay. So during daytime the stomata will be closed. Because of the transpiration rate will be reduced. Because of that, higher water use efficiency will be there. Clear? So during night time the stomata will be open. So during only night time the CO2 fixation will be happen. So this is the typical point about CAM plants. Okay. Example of CAM plant, if you see cactus, agave, pineapple, these are the some of the examples of CAM plants. Okay. This we have given here. The stomata will be open at night time only. That time CO2 will enter. Daytime stomata will be closed. Now we are going to see how the actual reaction or how the actual mechanism will happen. In night phase, what it will happen? In night time, CO2 will come. That CO2 will be fixed into malate. That malate will be stored in vacuole. This is another important question. Vacuole is the storage site for malate during night time. Okay, this is another important objective question they have asked. Okay, this brief cycle I will show at the last. Okay, then during daytime, the malate will be decarboxylated. That CO2 will be utilized by Rubisco enzyme. That CO2 will be utilized by Rubisco enzyme. This everything we are going to see in this diagram. Okay, you see here in this diagram, this is the simplified diagram. You just see here. First, during night time, stomata will be open, CO2 will come inside, CO2 will be converted into bicarbonate. Bicarbonate, by the action of phosphoenol pyruvate carboxylase, oxaloacetate will form. This oxaloacetate will be converted into malate, then stored in vacuole. ठीक है, then stored in vacuole. ठीक है, vacuole is the storage site for malic acid. During daytime, what it will happen means this malate will be decarboxylated. Malate is decarboxylating. This CO2 will be utilized here. This CO2 will be utilized by Rubisco enzyme in chloroplast that will lead to formation of starch. I hope this is clear. The question what they will ask here is in C4 plants, C4 plants, in C4 plants, C3 cycle happened in, C3 cycle will happen or it will occurs in where? Bundle sheath cell, bundle sheath cells. But in CAM plants, C3 cycle is happening in mesophile cells. These are the confusing question they will ask. Okay? In mesophile cells, C3 cycle will happen. I hope this is clear for everyone. This is very important also. This is very important also. I hope this is clear for everyone. Okay? With this, we are completing the dark reaction. And last minor concept, photorespiration. I hope this is clear for everyone. Okay? So we have completed light reaction and what are the variants of dark reaction C3, C4 and CAM pathway. I already told this Rubisco enzyme has two different activity. One is carboxylase, another is oxygenase. Due to this oxygenase activity, photorespiration will happen. That how it will happen, I am going to say here. This is another repeated question from here. Every year they are asking question. Photorespiration, every year they are asking question. Just to understand this figure, that is highly sufficient. Okay. You see here, RUBP molecule is there. If CO2 will come here, so CO2 plus RUBP. Okay. If this is happen, means 6 carbon will form. This will be lead to formation of 3 PGA. If oxygen is there with RUBP, that will lead to formation of two compound. One is PGA will form. Then second compound is 2-phosphoglycolate. 2-phosphoglycolate will form. This is clear. This is 3 carbon, this is 2 carbon, total 5 carbon was formed. I hope this is clear for everyone. If oxygenase activity is there, means the RUBP will be converted into, you just see here, this will be converted into 3-phosphoglycerate and 2-phosphoglycolate. That's why we are saying this as glycolate cycle. Glycolate cycle. This can also called a C2 cycle because this is a 2 carbon compound formed, C2 cycle. You shouldn't confuse that with glyoxylate. Okay, glyoxylate is different. This is involved for conversion of fatty acid to sugars. Okay, this is different cycle. So glycolate cycle is also known as C2 cycle. So 2-phosphoglycolate is formed. How this 2-phosphoglycolate is regenerated to 3-phosphoglycerate, that is known as C2 cycle. The question what they will ask means what are the organelles involved in photorespiration? You just have to remember CPM, chloroplast, peroxisome and mitochondria. These are the organelles involved. You have to know the sequence also. That's why I am writing CPM. First is chloroplast, second is peroxisome, third is mitochondria. And second most important question what they will ask means, second most important thing, in which organelle carbon dioxide and ammonia will be released or produced. Okay. In mitochondria, these things will happen. First, we will see the cycle. So, the 2-phosphoglycolate, if this phospho molecule will be released means that will lead to form of glycolate. This glycolate will come to peroxisome. It will be converted into glyoxylate. This glyoxylate will be converted into glycine. This 2 molecule of glycine will come to mitochondria. If you see here, a molecule of ammonium and CO2 will be released. That will lead to formation of serine. This serine will be regenerated to 3-phosphoglycerate. From this, we are very sure the mitochondria is the major site for CO2 and ammonia releasing. Okay, these are the key questions they are asking every year. I hope this is clear for everyone. You just go through this cycle. Then another important thing, superoxide molecules will be formed. Superoxide, hydrogen peroxide will be formed in peroxisome by the action of catalase that will be degraded there. Okay. So this is the important about C2 cycle. With this, we are completing photosynthesis and photorespiration. I hope no doubts are there. Any doubts is there? Kindly let me know. Any doubt is there? Any doubt is there? If no other doubt means we can have one break for 5 minutes, then we can proceed the remaining things. Kindly let me know any doubt is there. Safura, Surapsi, yes, no doubt, sir. Yes. We can resume at 12:18. Okay. After 5 minutes, we can see. Huh? After 5 minutes, we can see. Shall we start, students? Hello, hello, whether I'm audible? Hello, kindly reply whether I'm audible. Yes, yes, yes. Today we are going to see about mineral nutrition. Okay? Yeah. From mineral nutrition, sure we will expect 1 or 2 questions. Okay? Before going to the session, first I will tell what are the questions they will ask. Okay? From mineral nutrition, the major questions they are focusing. If you see the previous year question papers, the major thing they are asking is deficiency symptoms. Okay? Where the deficiency symptom will happen, whether it's an younger leaf or in older leaves. Then the most important thing what they are asking is, if a mineral nutrition is there, what is the major function? What is the major function it will be involved? Okay? So these things we are going to cover here. Okay? First we will see what is meant by mineral nutrients. Okay? What is meant by mineral nutrients? Okay? So the green plants are autotrophs. This we have already seen. They can synthesize their own food via photosynthesis. This we have already discussed. They will use carbon dioxide from air, water and mineral nutrients from the soil. Okay? Now we are going to see what is meant by minerals. Minerals are what? They are naturally occurring inorganic solids. This is most important. It's an inorganic solid having definite chemical composition and ordered atomic structure will be there. Okay. The study of mineral nutrition, we will know. The study of mineral is known as mineral nutrition. This is just for your understanding only. The key point you have to remember from this slide is minerals are inorganic ions. This you have to understand properly. Okay. If you see the classification of elements, mineral elements, we can classify broadly into essential element and non-essential element. Okay. We can broadly classify it into essential elements and non-essential element. Now we are going to see why we are saying some of the elements as essential and why we are saying some of the elements are non-essential. If we are saying essential element, there should be some criteria should be there. Okay. This is the question they asked in JRF 2021. Okay. The criteria for essentiality of nutrient was given by Arnon and Stout in the year 1939. This year also very important. 1939 he has given. Total 3 criteria are there. What is the criteria? The plant cannot complete its life cycle without the element. Without that element, the life cycle of the plant will not be complete. And the most important thing, the function of element cannot be replaced by another element. For example, nitrogen is there means that function will not be replaced by another element. Okay. Then the most important thing, the elements should directly involved in plant metabolism. These are the 3 essential criteria given by Anand and Stout for essential elements. Okay. If you see the essential element, there are total 17 essential elements are there. Okay. Total 17 essential elements are there. Okay. If you see the basic elements are carbon, hydrogen and oxygen. This they are coming from water and air. And the most important thing, the essential element are classified into 2 classification, that is macronutrients and micronutrients. These examples are very important. Okay. NPK, calcium, magnesium and sulfur is macro element, and micronutrient is iron, manganese, zinc, copper, boron, molybdenum, chlorine and nickel. This you have to remember. Okay. Apart from this essential element, there is non-essential or beneficial elements are there. Here you have to remember only the example. Okay. Non-essential or beneficial elements may be required by some plants for specific condition. For example, silica will be present in rice seeds. Correct. Silica will be present. That will give additional benefits. Okay. Fungal resistance will be there. Bacterial resistance will be there. Because of that, silicons will be present on the surface of the rice leaf. Okay. So example of this beneficial element is what? Silicon, sodium, selenium, aluminium, cobalt, vanadium and gallium. These are the example of beneficial elements. Okay. We have already seen the essential element are classified into 2 types, macronutrients and micronutrients. This macronutrients, NPK, calcium, magnesium and sulfur. Okay. If you see this macronutrients is further classified into 2 types, that is primary element and secondary element. So primary element is NPK, secondary element is calcium, magnesium and sulfur. Okay. Okay, so macronutrient means what? The concentration or the requirement will be more than 1000 mg per kg of the dry matter will be there. These are the macronutrient, and micronutrient means less than 100 mg per kg will be there. Okay, these are simple things only. Okay, so on the basis of general functions of an essential element, they have classified into different types. From here, sure they will ask one question. This is very important. Okay, what are the major function? For example, nitrogen, this will be primarily found in proteins and nucleic acid. Then sulfur, this is very much important for sulfur containing amino acid biosynthesis, particularly cysteine and methionine. Apart from that, phosphorus, key element in nucleic acid and ATP. Magnesium, it will play center role in chlorophyll molecule biosynthesis. And calcium, it will be involved in cell wall. This is the most important objective thing. In calcium pectate, calcium will be found. That will be found primarily in middle lamella of the cells. Okay, that will give rigidity to the cells. Okay. Then osmotic regulation. If you see the osmotic regulation, potassium and chlorine ions plays major role in osmotic regulation. Based on the osmotic regulation, this will be helps in stomatal movement, nutrient transport and cell turgidity. Okay. Apart from that, balancing or antagonistic elements are there. Some elements will counteract the toxicity of other element. If a particular element concentration is more, this element will accumulate to counteract the toxicity. For example, calcium can reduce toxicity of sodium. Okay, calcium can reduce toxicity of sodium. Another important classes is catalytic function. Okay, it can be act as a cofactor or coenzyme for different enzymes. You see this iron will be primarily involved in electron transport, particularly cytochromes will have iron content. Copper will be involved in redox reactions. Zinc for oxygen biosynthesis cofactor. This they have asked one time. Okay, zinc is the cofactor required for oxygen biosynthesis. Molybdenum for nitrate reduction. Manganese for water splitting or photolysis of water. Manganese along with chlorine. Okay, these two are involved in photolysis of water. These are the general classifications. Okay, general classification based on the major functions. Okay, now we are going to see total 17 elements are there. In which forms the plant will be absorbed? This is another important question. Okay, for example, sulfur will be uptaken in sulfate form, SO4 2 minus. Phosphorus will be uptaken in orthophosphorus form. Okay, H2PO4 minus, HPO4 2 minus. This is the most important thing. Okay, phosphorus will be uptaken in orthophosphoric form. Magnesium, calcium, potassium, nitrate, it can be uptaken both in nitrate and ammonical form. Oxygen, CO2, water for carbon, hydrogen and oxygen form. And another most important thing, one time they asked in which form the boron will be uptaken, boric acid form. Okay, BO4 minus. Okay, that question they have asked one time. Okay, these are the different forms. You just go through once. Okay. Now, the another important thing, this next from this next two slides, you will get one question. I will assure you. Okay. So, now we are going to see the mobility of the element. If the 17 elements are there, what are the elements that are mobile in plant and mobile in soil? Okay. So, you have to read the question properly, whether they are asking mobility of the element in soil or in plant. First, we will see about soil. Okay. In soil, boron, chlorine, nitrate and sulfate. These four are the only nutrients that are highly mobile in nature. Okay. Boron, chlorine, nitrate and sulfate. These are highly mobile. This is very important question. Then immobile, remaining all are immobile in nature. Then in plant, if you see, the mobile element are NPK, chlorine, magnesium, molybdenum and chlorine, molybdenum and magnesium. These six are mobile element. Immobile element means phosphorus, calcium, copper, iron, manganese, zinc and sulfur. Okay, this is very important. Why I am saying means they asked one time question. Which element is immobile in soil but mobile in plant? Okay, which element is immobile in soil but mobile in plant? If you see phosphorus, phosphorus will be immobile in soil but mobile in plant. This is very important question you have to remember. Then another most important thing, If a element is mobile in plant, the deficiency symptom will happen in older leaves. Okay, mobile means the deficiency symptom will happen in older leaves. If it is an immobile element, the deficiency symptom will happen in younger leaves. This is another important point. So if we know which element is mobile or immobile in plant, based on that we can easily filter out the options in question. Okay, whether the symptom will happen in younger leaves or older leaves. This is the repeated question they are asking. Okay, if a mobile is immobile, if an element is immobile, where the symptom will happen, and what is the example of that mineral element. Okay, this is the another important thing. Nutrient deficiency symptom classification. Okay, if older leaf symptom is happening means ultimately it's a mobile element. Okay, so these are the mobile element. If the veins are green, that will due to magnesium ion. If dead spot are there means that is due to potassium and molybdenum. If no dead spot are there means nitrogen, phosphorus and magnesium. And another most important thing, from here you will get one question. Okay, repeatedly they are asking, if iron deficiency is there means deficiency will be always happen in new leaves, that is intravenous chlorosis will be happened. Okay, if magnesium deficiency is there means intravenous chlorosis will be happen in older leaves. This question they have asked. Clear? So this is very important chart, you have to just remember. Okay. Now we are going to see each and every element. Separately we will see nitrogen, phosphorus, every element. What are the key points you have to remember in exam? That are only we are going to discuss. Just 5 minutes. Quickly we are going to revise. Okay. So nitrogen, we know the absorbed form will be nitrate and ammonia. Okay, rarely nitrate form will be there. The major function is what? It will be involved in component as proteins, nucleic acid, chlorophyll, enzyme, alkaloids and vitamins. Deficiency symptoms, chlorosis will be happen in older leaves, and the most important thing, anthocyanin pigmentation will be there in stem and veins. Okay. Then if the deficiency is there means stunted growth will be there. These are the key point. Phosphorus, we know uptake form will be the H2PO4 minus or HPO4 minus. It is important for ATP and nucleic acid structure, energy transfer reaction it will be involved. The most important thing, the deficiency symptom, what it will happen means, this is the most important. Okay. Dark blue green leaves will be there. Dark blue green leaves will be there. Apart from the premature leaf fall and necrotic spots will be there. If you see about sulfur, we already discussed sulfate form it will uptake, involved in cysteine and methionine biosynthesis. These are the key points. Then if you see the deficiency symptom, pale yellowing of young leaves, this they will ask. Okay. Pale yellowing of young leaves will be happen due to sulfur deficiency. This is another important point. And most important thing, inward curling will be there. Inward curling of tips and margins will be there. These are the important points you have to remember for exam. Okay. Next we are going to see about calcium and magnesium. Calcium uptakeal form will be Ca2 positive. This we have already discussed. It will be primary component in middle lamella. Calcium pectates will be there. If deficiency symptom is there, this is JRF 2023 question. If deficiency symptom is there means disintegration of meristematic and young tissues will be happen. This is JRF 2023 question. Okay, meristematic region of the tissue will be affected. Okay. Then magnesium, we know magnesium Mg2 plus form it will uptake. The most important thing is central atom of the chlorophyll will have this. Then the deficiency symptom is intravenous chlorosis in older leaves. If intravenous chlorosis is in younger leaf means that will be iron. Okay, this is the key point you have to remember. Okay. With this, we are completing the macronutrients. Then micronutrients. Simply, we are going to discuss the key points. Okay. If you see iron, uptakeal form will be Fe2 plus and Fe3 plus. Low mobility in the soil will be there. In plant will be there. This we have already seen. Deficiency symptom, if you see, intravenous chlorosis of young leaves will be happen. This is the key point you have to remember. Deficiency symptom means intravenous chlorosis of young plants will be there, intravenous chlorosis of young plants. Next thing is manganese. This will be involved in photolysis of water. This we have already discussed. Okay. The major deficiency symptom is gray speck. This is another important question they have asked. Okay. They asked in match the following. Okay. Gray speck of P is caused due to deficiency of which mineral nutrient? This is due to manganese. Okay, this is another important point you have to remember. Zinc, this we already discussed. Zinc will be primarily involved in auxin biosynthesis via tryptophan. Okay, deficiency symptom, clustering of young leaves. That means what? Rosetting. Rosetting will be happen. This is very important point. Okay, zinc will cause rosetting of the leaves. Rosetting of the leaf. This here we have given crop specific symptoms. This they can ask in general agri question also. Kyra disease of paddy is caused due to deficiency of which mineral nutrient? That will be caused due to deficiency of zinc mineral nutrient. Okay. Now we are going to see about molybdenum. Molybdenum, whiptail of cauliflower is caused due to molybdenum. This is the deficiency symptom, and the major function, if you see, nitrate reductase and nitrogenase enzyme will have this molybdenum in its structure. Okay. Next thing is chlorine and nickel. This all like very simple only. The major thing, chlorine, photolysis of water, it will be involved. If chlorine is deficiency is there, means bronzing of the leaf will be there. Bronzing of leaf will be there. Nickel, this will be primarily involved in urease. Urease cofactor is nickel. This I have already discussed in our lecture series itself in biochemistry class. Okay. Nickel, urease enzyme will have a cofactor. Then for urea metabolism, this nickel will be highly important. And last important thing is boron. Boron is very much important for sugar translocation. This is the key point, sugar translocation, and most important thing is pollen germination and pollen tube growth. These are the key points you have to remember. Okay. Deficiency symptom, what it will happen? More important one. Okay. Hollow stem in sunflower. Okay. Hollow stem. Brown heart. Internal cork in apple. Cracking of stem and fruit. These are the typical symptoms. Fruit cracking. These are the typical symptom for boron. You have to remember this. Okay. These are the key area. Whatever we are taking given in the slide, this is highly sufficient to cover the mineral nutrition portion. So you just revise this properly. Okay. Then the last element, copper. Okay. Copper, the most important thing, plastocyanine. This we have already seen. Copper containing electron transport chain. Okay. That will be there. Copper containing element. Okay. This will be involved in lignin synthesis also. This will be involved. Okay. The major deficiency symptom, if you see, leaf tip dieback will be there. This is the major deficiency symptom. Or dieback of shoot tips will be there. This is the major deficiency symptom of copper. With this, we are completing the mineral nutrition. I hope this is clear for everyone. Okay. What is mineral nutrient? We have seen what are the classification of mineral nutrients. Okay. What is the major function of each and every mineral nutrient, and what are the major deficiency, and what is the mobility of this mineral nutrient in plant as well as in soil. I hope this is clear for everyone. Any doubt is there in mineral nutrition? Just let me know any doubt is there. Kindly let me know any doubts are there. Any doubts? Yes, no doubts. Yes, if no doubts means we can proceed. Okay. Last two topics are there. One is photoperiodism and from vernalization. Okay. And again I am saying from here sure you will get one question. From photoperiodism every year they are asking one question, and from vernalization every year they are asking one question. I have compiled the important things only. This is highly sufficient to cover that topic. Okay. So we are going to see photoperiodism. In this question what they will ask means example for each and every crop. What is short day plant, long day plant, day neutral plant, like this examples they will ask. Okay. This is the most important thing. Then they will ask the term photoperiodism was given by whom. Okay. Then another most important thing, in short day plant, whether day period is critical or night period is critical. Like these questions only they will ask. Okay. That all the things we are going to see now. Okay. First, what is meant by photoperiodism? First, we will say what is meant by photoperiod. Okay. Photoperiod means the relative length of day and night is known as photoperiod. Okay. The relative length of day and night is known as photoperiod. Okay. So, what is meant by photoperiodism means the response of plant to photoperiod, that is the relative length of day and light required for flowering. Okay. For flowering is known as photoperiodism. Okay. The term photoperiodism and the thing was discovered by Garner and Allard. This is JRF 2023 question. Okay. JRF 2023. Okay. They asked the question in match the following. Garner and Allard given the term photoperiodism. Okay. TD Lysenko. TD Lysenko given the term vernalization. Okay. Vernalization term was given by TD Lysenko. These both question they have asked. Okay. So Garner and Allard for photoperiodism, TD Lysenko for vernalization. Okay. Now the plants are classified into three different types based on the relative length of day and required for day and night required for flowering. Okay. First is short day plants, second is long day plant, and third is day neutral plant. You have to remember the example for each and every plants. Okay, short day, long day, and day neutral. And most important thing, which is the critical period required for flowering in short day and long day plants. Okay. So short day plants means what? These plants requires relatively short daylight. Okay. The daylight should be less, usually 8 to 10 hours should be there. Okay. Then continuous dark should be there. Okay. They need a continuous dark period of about 14 to 16 hours for flowering. So we can call this short day plant also as long night plant. Short day or long night plants. Okay. These examples are very, very important. Okay. So the short day plant example are what? Marymouth variety of tobacco, biloxi of soybeans, cochlear of Xanthium pennsylvanicum. Okay, these are the typical example. And the critical period for short day plant is what? The dark period is critical period. Okay. So the dark period must be continuous. If it is even interpreted briefly by a red light, it will inhibit the flowering. Okay. So if we give the red light, it will inhibit flowering in short day plant during the dark period cycle. Okay, clear? So the critical period for short day plant is dark period. Next question we are going to see about long day plant. Okay, long day plant we can also call as short night plants. Okay. The recruitment if you see, these plants requires long daylight period. Okay, daylight period should be long. Usually 14 to 16 hours will be required. Usually 14 to 16 hours period of long day condition is required. The typical example is what? Henbane. This they have asked in JRF 2020, I think so. 2020 or 21 I forgot. But they asked this one. Henbane is the example for long day. Spinach and sugar beet is also an example for long day plants. Okay. The critical factor is in long day plant is light period. Light period is the critical. Okay. If we expose a brief, if the light period is interpreted, flowering may be inhibited. Okay. So this is the critical point. Okay. Next is day neutral plant. Okay. Day neutral plant and intermediate plants. Okay. So day neutral plant means what? Flowering occurs in all photo period ranging from 5 to 24 hours. The examples are very, very important. Okay. The example are very, very important. Tomato, cotton, sunflower, cucumber, and certain peas and tobacco are critical example for day neutral plants. I hope this is clear for everyone. Okay. Further, the short day, long day, and day neutral plants are classified into different things. What is that? Long short day plant and short long day plants. Okay. So long short day plants means these are actually a short day plants, but they requires long day condition during yearly period of growth. I hope this is clear. This is actually a short day plant, but it requires initial long day periods for flowering. Okay. Then short long day plants means it's a long day plant, but it requires short day period during yearly periods of growth. And the example we have to remember long short day plants example is bryophyllum, and short long day plant example is wheat and rye. Okay, wheat and rye are example. Okay. Another most important thing, I have classified all these things in an exam ready quick revision table. Just you have to remember this one. Short day plant, short day should be 8 to 10 hours should be there. Long day means light should be 14 to 16 hours should be there. It can flowering at any photo period. Day neutral means. Okay, night requirement they have given, okay, also called as what, and the critical examples I have given here. You have to just revise this quick revision. Okay, clear students? This is quick revision for everyone. Okay. Next thing, if a photoperiodic stimulus is there, which part of the plant will perceive that stimulus? Okay, if flowering stimulus is there, whether that will be pursued by what organelle, whether it's a root, shoot or leaf, what is the organelle that will perceive that, what is the part of the plant that will perceive the stimulus? That is very important. Okay, you shouldn't confuse here. Okay, so the photoperiodic stimulus will be perceived by leaves. I am writing here itself, photoperiodic stimulus will be perceived by leaves. Okay, vernalization or cold stimulus will be perceived by apical meristem. You shouldn't be confused. This they can ask in statement question. Okay, statement they can give. Okay, so vernalization or cold stimulus will be pursued by apical meristem. Okay, photoperiodic stimulus will be pursued by leaves. Okay, the hormone involved for flowering we can also known as floragen. Okay, floragen hormone will be involved for floragen. Okay, for flowering. Okay. Okay, next thing is vernalization. I hope this is clear. We have discussed what are the important points. Photoperiodism means what? What are the classifications? Okay, what are the typical examples? Now we are going to see about vernalization. Okay, what is vernalization and who has given the term? Yeah, what is vernalization and who has given the term? That we are going to see. Okay, so vernalization means what? Vernalization means it's a acquisition or acceleration of ability to flowering by chilling treatment. This is the definition. You just have to remember the definition. Okay, that means we are giving the cold treatment for initiating the flowering, that is chilling treatment. Okay, the term vernalization was given by T.D. Lysenko. This I already told. Torifim Denevos Lysenko. That is T.D. Lysenko. He is a Russian genetist. This is very important point you have to remember. Okay. Then another important point. This line they ask directly as one question. Okay. Beside photoperiod, certain plant requires low temperature treatment during early life stage for subsequent flowering is known as vernalization. You shouldn't confuse this term with stratification and scarification. You know, stratification means cold treatment for breaking the dormancy. Clear? That is stratification. Stratification. I will write areas. Tartification. Okay. Stratification means cold treatment for breaking dormancy. Okay. For breaking dormancy. Clear? You shouldn't confuse this with vernalization because they have asked this question. In option they have given stratification also. Okay. So the key principle is what? We are giving the low temperature treatment at the early stage of the life for improving the flowering. Okay. This is the important thing. Okay. Next important thing, what are the major, then where the perception of the cold stimulus will happen? This we have already discussed. The perception site is what? Apical meristem is the perception site. Okay, the cold stimulus is perceived by apical meristem. Okay, apical meristem is the site for perception. Okay, then for flowering, which hormone is involved? That we also seen. Floragen. Okay, floragen is the hormone involved for flowering. This they have further classified as vernalin. Okay, for vernalization, vernalin hormone will be involved. Like that they have given. Okay. Like that they have given. Okay. Now we are going to see the major conditions required for vernalization. Major conditions required for vernalization. Okay. So the age of the plant, this is like differ based on the crop. The most important thing you have to remember is the temperature required for vernalization. Okay. Most commonly we will use 1 to 6 degrees Celsius. This is most important thing. And another most important thing is oxygen and aerobic process. It's an aerobic process. Okay, aerobic process, oxygen should be required. Okay, and sufficient hydration of the seed also should be there. Another most important, these are the important things. Okay, now we are going to see the brief of today's class. Okay, I hope no doubts are there still now. First I will I will give a brief outline what are the things we have discussed. Okay. First we start with photosynthesis. We have seen the definition of the photosynthesis. Okay. In photosynthesis, we have seen about the history, how the things will happen. Okay. Who has discovered the thing. Then we see about plant pigments. All the things are going in your mind now. Plant pigment we have seen, its classification we have seen. Then how the light reaction, how the light will be absorbed by the chlorophyll molecule, how the light reaction will happen. Okay. Then how the dark reaction will happen. Okay. Here we have seen C3, C4 and CAM pathway. Okay. Then at last we have seen C2 or photorespiration cycle we have seen. Due to what? Due to oxygenase activity of rubisco. Okay, with this we have completed the photosynthesis topic. Then at second we have seen about mineral nutrition. Okay, in mineral nutrition we have seen the classification of minerals, the major function of each and every mineral and its deficiency symptom. At third section we have discussed about photoperiodism. Okay, we have discussed what is meant by photoperiodism, who has given the term photoperiodism, and at last we have classified the photoperiodic plant into short day, long day and medium short day, long day and day neutral plants. Then we have seen the example for each and every crop. Then at last section we have seen about vernalization. Okay, vernalization, who has given the term and what is the process. Any doubt is there, students, till now? With this, we are completing our lecture. Any doubt is there? Any doubt is there? No, sir. Yes. Kindly post your valuable comments on the videos after this session. Okay, whatever improvement we have to done from our side, kindly let me know in the chat session. Okay. Kindly don't forget to mention your suggestions and doubts in chat session. Okay. Kindly mention. Okay. That will be useful for us also. Okay. Your valuable suggestions are needed. Yes. Yes, thank you students, thank you. We will see tomorrow. Okay, tomorrow same timing 11 to 1:30. Okay. Tomorrow we are going to see the most important topics. Okay, we are going to see phytohormones. What are the different plant hormones that we will see? Apart from that, respiration. Respiration we are going to see completely from A to Z of respiration we are going to see. Apart from that, stress physiology. These three are the topics tomorrow we are going to see. Don't miss the class, kindly join. Okay, thank you students.